Hi everyone, I'm Colby Agostino.
And I'm Olivia Schmitt.
We did research last semester under Dr. Corneli in the Department of Chemistry and Biochemistry. Our project is called the exploration of K2 mycobacteriophages with BRED. BRED stands for bacteriophage recombineering with electroporated DNA, and we'll get into what that means in a little bit here. So before we begin, it will be useful for us to talk about what a mycobacteriophage is. So just in general, phages are the most abundant biological entity on the planet. And then specifically, mycobacteriophages are viruses that are capable of infecting mycobacterium. Mycobacteriophage are organized into clusters and subclusters. Like I said earlier, we're going to be working with K2 phage, which are actually, they're able to infect Mycobacterium tuberculosis, which is the bacteria that's responsible for causing tuberculosis.
So why are phages important? In the age of antibiotic resistance, phage therapy offers a promising alternative to antimicrobials. Mycobacterium tuberculosis, as Colby mentioned, the bacterial pathogen that causes human tuberculosis, is one of the most dangerous antibiotic-resistant bacteria. Our lab works with the non-pathogenic version of tuberculosis called M. smegmatis. In addition, Mycobacterium abscessus is another distantly related drug-resistant mycobacteria that causes severe lung infection that we'll be taking a look at in a few slides.
All right, before we talk about our project, it'll be useful just to have some background knowledge about the life cycles of phage. So there are two possible life cycles phage can participate in, the lytic and temperate life cycles. As you can see in this diagram on the right, they both begin with the phage binding to a receptor on the bacteria and inserting the phage DNA. Then in the lytic phage cycle, the phage DNA uses the bacteria's machinery of the cell to reproduce itself, and this eventually results in the lysis of the cell, which is equivalent to cell death. But then in the temperate life cycle, the phage DNA actually becomes integrated into the bacterial genome, and this is preserved through multiple rounds of binary fission. And then the genes responsible for the temperate or lysogenic cycle on the right here are the integrase and immunity repressor genes. And that's something important to remember because that's what our project will be focused on.
So something really exciting that happened this past May was that a combination of 3 phages were utilized to successfully heal a cystic fibrosis patient in the United Kingdom infected with Mycobacterium abscessus. And one of those 3 phages, ZoeJ, was actually isolated on Providence College campus in 2012. More importantly, this was the first instance that genetically modified phages were used to treat a bacterial infection, which is why Colby and I would like to direct our research to replicating this method whereby the phages were genetically modified in order to help phage therapy and serve the medical community. And located on the pictures on the left side, we have the patient Isabel, who was successfully treated, and her mother, as well as Graham Hatfull at the University of Pittsburgh. And beneath, we have the 3 phages that were actually used to treat Isabel.
All right, so now we'll talk about the chief objective of our research is to produce 2 mutant strands of mycobacteriophage that we have on Providence College campus. And the goal of this mutation is so that both of these phage will behave lytically and destroy the host bacterium. Now Olivia will tell you a little bit about her project.
Yeah, so I'm working with the phage Marcoliusprime, as Colby mentioned, a K2 subcluster phage capable of infecting Mycobacterium tuberculosis. My goal is to delete the immunity repressor because, again, the repressor allows the phage to maintain lysogeny. So if this gene is deleted, then we can hopefully help the cell enter the lytic cycle only.
And I'll be using a mutant strain of ZoeJ, the same mutant strain that Dr. Corneli knocked the repressor out of and that we discussed in the last slide that was used to treat that UK patient. I'll be knocking the integrase out of this mutant strain, so we'll effectively have a double mutant with the hope that it will increase the phage's ability to infect and kill bacteria and be used as a viable option for phage therapy. And how we will accomplish obtaining these mutant phage are through BRED, which we talked about on the first slide, is bacteriophage recombination of electroporated DNA. It's a technique used by the Hatfield Lab, and we'll discuss what that means right now.
So the first step of BRED that Colby and I accomplished in the fall was to design a substrate or G block. So what is a G block? It's basically a 400 base pair double-stranded DNA substrate with 200 base pairs of homology, both upstream and downstream of the gene that we'd like to delete. In my case, the immunity repressor, and in Colby's, the integrase. So when the allelic exchange reactions occur, as you can see in the diagram on the right, the reaction occurs without the portion in the middle because there's no DNA homologous to it. So Colby and I also designed primers to amplify this G block that we designed in order to obtain more of it, which Colby will show in the next slide.
Yeah, like Olivia just said, we only received a very small concentration of the G block. So we had to amplify this using a common laboratory technique called PCR. PCR is useful to amplify nucleic acids. And as you can see in this diagram on the left, this is a result from gel electrophoresis after the PCR, which was accomplished with primers that we designed ourselves. And as you can see, these bands in lanes 2 through 5 are about 400 base pairs, which corresponds to the 200 base pair of upstream and downstream homology that Olivia was just talking about.
So our most pressing and definitely most difficult task this semester was to obtain electrocompetent cells, which Colby and I were actually able to do before the start of spring break. So electrocompetent cells are necessary to ensure that the bacterial cells take up the phage DNA and the substrate that I mentioned before. So Colby and I prepared liquid cultures from ice crystals of the recombineering strain Mycobacterium sphagmatis plus the PJV53 plasmid. So what is this plasmid? PJV53 contains a gene that encodes for kanamycin resistance, which will allow us to select for bacteria that have this recombineering plasmid. More importantly, PJV53 actually has the recombineering enzymes necessary to accomplish these allelic exchange reactions. So Colby and I, like I said, created the liquid cultures. When the optical density reached 0.4, optical density is just a measuring of cell scattering. The acetamide was added, which turns on the recombineering genes.
All right, so the next step of BRED would have been electroporation. Unfortunately, we didn't get to this step of BRED because our semester was ended short, but hopefully we'll be able to continue our research and complete this step either over the summer or next fall. And what would happen under this electroporation step is that we'll use the electrocompetent cells that Olivia was just talking about, the amplified substrate that we mentioned earlier in the phage DNA, and we'll apply an electric field to this solution. This will increase the permeability of the cell membrane so that the phage DNA and the substrate can enter into the cell. And then the electric shock will also facilitate the allelic exchange reactions so that we'll have mutant phage genomes. And then after a short recovery period, we'll do multiple rounds of purification and plating. So as you can see in the diagram, on the right side of the diagram, we have a plate that's covered in a nice bacterial lawn. And each of those little black circles is actually a plaque where phage is present, and hopefully some of those plaques will contain the mutant phage that me and Olivia are trying to create.
So lastly, as Colby mentioned, we have plaques on the bacterial lawn, and while some of the plaques contain wild-type phage DNA, only some of them will have the phage mutant. So in order to detect which plaques have this mutant, we will perform a series of PCR screenings, serial dilutions, and plating on the lawn. The amplicon for the mutant phage, when we perform the PCR experiments, will of course be shorter because it doesn't contain the gene that we knocked out. And once we detect which plaques contain the deletion, we'll actually purify with 3 rounds of purification, as Colby mentioned before. So hopefully we'll be able to do this either this summer or in the coming semester in the fall.